My name is Silke Dodel, and today I'm going to talk about artificial intelligence for information extraction from scientific publications. I'm the founder of Deep Science AI. Deep Science AI is a startup. You can visit us at our website at www.deepscienceai.com. Deep Science AI was lucky enough to be part of Venture Class 7 at FAU Tech Runway, where we received a lot of support and great mentoring. Today, what I will talk to you about what we do and what our mission is. So what we do is we build artificial intelligence systems for information extraction and problem solving. So kind of having a second brain. So we don't aim at replacing people or scientists or professionals, but we aim at giving them a kind of a computerized second brain with some reasoning ability, but greater capacity. So our mission is accelerating scientific discovery by building artificial intelligence systems that reduce information overload and help navigate complexity. The outline of my talk today is as follows. So first, I will go over the problem. Literature search information overload. So information overload is a big problem for all of us, and it's especially pertinent when doing some literature search. Every year, thousands of articles are published, and researchers are often overwhelmed in keeping up even in their own narrow field. So we solve or aim to solve that. So what we built is an artificial intelligence system that we call Archimedes System, and that extracts information from a large number of publications. I will present the Archimedes System in form of an example, and then I will shortly review the features of the Archimedes System and give you some ideas of the use cases for the Archimedes System. And then we can have a discussion. If you're at the live presentation, you can shoot me some questions. If not, you can always contact me through our website or by email. Okay, so what's the problem? Information overload slows down science and business development. So if you are a biomedical researcher or really any researcher, this image may look familiar to you. You have an ever-increasing pile of publications, either as actual papers on your desk or as files on your computer that you should read to keep up with the latest development. And if you're working in the biomedical fields or any other field, actually, complexity is ubiquitous and affects everything from the correct interpretation of experimental results to finding the right treatment for a patient to identifying the right business strategy. So Deep Science AI can help with both complexity and information overload. So today we will focus on information overload and in the particular case of literature search. So traditionally, the literature search in the traditional way leads to information overload. So here's what traditional literature research often looks like. So you type in your search, you hit enter, and you are getting thousands of search results, which feels more like infinitely many search results. So you might start scanning the results. So you go through the search results one by one, you take notes, you save articles, you do that for result number 1, result number 2, result number 3, result number 4, and so on and so on. By the time you reach result number 100, you get completely exhausted and you might give up. So, but what if you stop too early and miss an interesting result? So that's where we come in. So this is why we built the Archimedes system. This is a system developed by a deep science AI for information extraction from a large number of scientific articles. We call the system Archimedes system for the eureka moment of Archimedes. So we hope that our system will facilitate a lot of eureka moments for its users. So what is the idea of the Archimedes system? The idea of the Archimedes system is to act as interactive filters. So let me walk you through this picture here. So you have your literature search using your favorite search engine, let's say Google Scholar, or for if you're a biomedical researcher, you might use PubMed. Then without the Archimedes system, you will get overwhelmingly many irrelevant results indicated down here, and you might not really know which results are relevant without scanning the results individually. Now, the Archimedes system is not designed to replace a search engine. That's very important. So what we are doing is the Archimedes system comes in after the search results and treats the search results. So you are still using your favorite search engine, but the Archimedes system is extracting the information from the search results, so you don't have to scan each result individually. So basically, we are taking a detour here. So the search results, we are typing in our search results, our search query. We are getting our search results. Those are fed into the Archimedes system, which acts as an interactive filter, meaning it's going to extract the information, represent them to you in a certain way. You can interact with it and identify your interests, and then it will, based on this interactive filtering process, you will be left with highly relevant results in a fraction of the time it would take you to scan those results. So let me go over an example. So this is an example of a biological researcher who had found some genes in the zebrafish, and he was interested in finding out more about these genes, which were overexpressed in some conditions. So you can here enter then your PubMed query. So he was interested in the BDNF, EGR3, NPAS, FOSB, and that in zebrafish. So you you click your search results. Then here you have 4 buttons under your search bar. One is query info, second article list, third topic map, and fourth main results. So I will go through each of them individually. So the query info is actually most useful for people who might not be so familiar with some of those genes, like let's say a Ph.D. student who got from his or her Ph.D. supervisor those genes and say, oh yeah, look those up. Well, the query info then tells you something more about the query that you have put in. Here, the query information extracts some information from the Human Protein Atlas. So it tells you the name of the genes. So for BDNF, that would be brain-derived neurotrophic factor, and it gives you the basic function. So this gene encodes a member of the nerve growth factor family of proteins. And so it does so for each entry. And if you're interested in having more information, you can click and it will bring you outside to the human brain protein atlas. So then the second button, if you hit that, you get the article list. So that is, if you use the Archimedes system like that, it's basically no different from using the search engine itself. You get a list of articles related to the query. Here we found 100 results. That's actually true. We exactly found 100 results, not more, not less. And we show the results a little bit different than usual. So here you can sort the results not only by date, but also by journal and by title. So by default, all results are taken into account, and you can unselect some of those results that you might not be interested in. But again, this is only the regular part. Now we are interested in actually going into those publications. And the first step is to create a topic map. So this is in this third button here. So the third button gives you a topic map. So the topic map extracts the topics in the articles that are related to the query. So here in this query, it extracts the following topics. So the main topics: metabolism, physiology, drug effects, toxicity, pharmacology, and genetics. And then you have some subtopics that are present as well. And here those numbers tell you how many articles are treating this topic. And as you can see here, by default, everything is selected, and you can unselect if you're not interested in some of the subtopic, and then you can also click on the numbers, and that brings you directly to the article. So this is already one filter, kind of a high level filter if you want to use this. Now, to dive in deeper, our system actually extracts the main results from the paper, and that's where this fourth button comes in. So if you click on main results, the system will show you a grouping of related results. And so those those groupings are over different publications. So instead of you going from one publication to the other and saying, oh, geez, yeah, five articles ago or so, I saw a similar result. So let me put that somewhere similar. The system does it actually for you. And then it will show you some representative sentences for each result group. So those 5 sentences that you see here are representative sentences for a group of results over different publications. And we show those results with pubtator highlighting. Pubtator is a variant of PubMed, which allows you to highlight some biological concepts in the scientific articles. So it currently covers 6 biological concepts: genes, diseases, chemicals, mutation, species, and cell line. However, it does so only within one article, but our system actually shows you those bioconcepts over several articles, and they are listed here. So here you have those 6 bio concepts that are present in those results, and then under those you have the list of um, specific entities. So here BDNF, not surprisingly, shows up very often. So here it shows up 214 times. That's actually more than the search results. That is because it actually counts multiple occurrences in those articles. So don't be surprised. And now again, here you could actually remove some of those entities if you're not interested in them, or if you click on those entities, you will be brought to the human protein atlas for more information about these genes as well. So we have those five sentences. Now you can look over those 5 sentences. So the first sentence talks about where BDNF is found in zebrafish. The second sentence talks about a specific chemical, fenformin, and its efficacy against radiotoxicity. Third sentence talks about locomotor hyperactivity and its relation to levels of calcium and sodium ions. Fourth talks about a specific expression pattern of a gene in the zebrafish brain. And the fourth talks about changes in neuroglia, neuroinflammation, and apoptosis, and especially epigenetic modulation in stress responses in zebrafish. Now, the biological researcher actually had found this one most interesting because he wasn't aware of any epigenetic relationship of these genes that he was researching. So, so if you're specifically interested in some of those groups of results, you can then choose those. So he would choose this one. You can obviously choose more than one. So when you do that, you can browse and select the most similar sentences in this group, and then export the selected groups of related results. And keep in mind, those are results over different publications. So here this is the selected sentence that we have seen before. And then, so in the main results, it's only one representative sentence per result group. So not to overwhelm you. Then once you have identified one of the result groups as a result interest, you can select this and then look into that further and look at the most similar sentences. So, so here we we see here that the a similar sentence is a zebrafish model of stress, and it has similar genomic responses. And the second one is it talks also about modulation of microglia, astrocytes, neuroinflammation, apoptosis. So it's quite similar to the selected sentence. And you can, just as before, you can select or unselect sentence. So by default, all the sentences are selected, and you can unselect some sentences that you're not interested in, and you can then export or save those groups. So the good thing is then that you then have with your account, you have some safe searches, so you would actually already have a list of sentences along with their references that are of interest to you, and along with a list of bioconcepts that are present in those sentences. So this helps you get a high-level overview of results in quite many publications. And of course, then at the end of the day, you would still look at the actual publications themselves, but you didn't have to go through each publication one by one. So let me wrap up the features of the Archimedes system again. So the Archimedes system combines multiple sources of information, literature, and ontologies. So ontologies are a selection of entities in a field and their functions. So for instance, the human protein atlas is an ontology which lists all the proteins and genes that we know of, and their relationship. Then the Archimedes system extracts and groups contents from inside the publication. So you don't only group from the title or so, it actually moves inside the publications and groups related results together. It helps you select and refine the most relevant results. So you are in charge. The system actually shows you what it finds related together, but you are in charge of choosing. Okay, this is of interest. This is not of interest. And the system eventually will learn what to present you over time. Then you can save and export your search, so you can also come back to a safe search, and you will have the whole hierarchical structure that I went over, and keep looking at it, see if there were some new developments. So let's say you're writing a paper and you did something else, and then after 2 months you revisit the search, then the system will actually alert you to some new things that have been going on since then, and relate them to the results that you have already saved. So use cases of the Archimedes system, obviously report writing. So you export your searches with the references, and you already have a draft of the literature review part of your report. So with the sentences that you know are important in this article, and then the reference list at the end. So you basically can just copy and paste that. Of course, you still need to write the report yourself, including the literature review, but it greatly accelerate this process. Then it also can help you in interdisciplinary research. So everybody is very fond of interdisciplinary research, and it's a very important development. The problem is that oftentimes when we talk about interdisciplinary research, basically what's happening is that researchers from different fields work together under the same roof, but they barely talk to each other because we have very different jargons in our fields. We have very different processes in our field, and it's kind of hard to understand sometimes why people are interested in something. My own background is computational, so I get all excited if I see if I see formulas. Now, if I see some particular biological entity, and the biologist is all excited about like an overexpression of this particular part somewhere, I don't really know what to make of this. And on the other hand, the biologist might go into a computational talk and kind of say, and so what does this formula help me? Now, neither of the researchers have time to actually read thousands of pages and basically do a second study. So the Archimedes system can help you get a quick overview of your colleague's field and learn how to understand their jargon. In the same vein, the Archimedes system can also help students learn new topics, and it can also help you create your own ontology, so of your own field, and refine them over several searches. So remember, you can save your searches, and so you can combine different searches from the past. So if you combine the searches from the past, you will actually see what kind of, for instance, concepts in biology came up most in your searches. So you might actually be able to identify some compounds or chemicals that you have not thought about before, to actually study more and create basically your own experience, your own ontology of what you are researching. And the good thing is the Archimedes system, being a computational system, it actually can be used for information extraction for any fields, because the mathematics behind the Archimedes system is applicable to any field, not only the biomedical field. So the Archimedes system is highly customizable. So this concludes my talk. So if you have questions, send them to me. If you're interested in a collaboration or partnership, we are always looking for collaborators and partners. Do you want your own custom-built Archimedes system? We actually offer building Archimedes systems for our clients. You can always send us a message on our website, www.deepscienceai.com, or you can contact us at archimedes.deepscienceai@email.com. Sorry, archimedes.deepscienceai@gmail.com. Thank you, and I look forward to talking to you. Thank you.